The information in this podcast is provided for informational purposes only. You should not use any information discussed in this podcast and related materials to make medical or healthcare related decisions. Always consult your physician or other qualified healthcare provider with regards to diagnosing and managing your medical condition. Any medications or treatments, including any discussed in this podcast, should be initiated and managed by a qualified healthcare professional. Welcome to Long COVID The Answers. I'd like to introduce Professor Nancy Klimas MD. Dr. Nancy Klimas has 40 years of professional experience and has achieved international recognition for her research and clinical efforts in complex chronic disease, notably myalgic encephalomyelitis stroke chronic fatigue syndrome, Gulf War illness, fibromyalgia, and most recently, long COVID. Professor Kilmas is a past president of the International Association for ME/CFS and a past member of the Health and Human Services CFS Advisory Committee, the VA Gulf War Illness Research Advisory Committee, the National Academy of Medicine's ME/CFS Clinical Case Definition Working Group, and has served on several NIH advisory panels. She is Professor Emerita at the University of Miami's Miller School of Medicine, and she is the Director of the Institute for Neuroimmune Medicine, Assistant Dean of Research, and Professor of Clinical Immunology at the Dr. Kiran C. Patel College of Osteopathic Medicine in Florida in the USA. She chairs the Department of Clinical Immunology at Nova Southeastern University. Professor Klimas founded the NSU Institute for Neuroimmune Medicine, where she directs a group of 18 interdisciplinary scientists and clinicians who are working to discover and implement innovative strategies that effectively treat or prevent complex chronic disease. Welcome, Nancy.
Thank you.
Do you have any
breathe for a moment after all that?
Well, it's just an amazing career. I think people need to know about it. So, Nancy, do you have any conflicts of interest to declare?
I do not. Thank you.
Excellent. So today I'd like to explore some of the features of your recent research paper entitled Remission of Severe Forms of Long COVID Following Monoclonal Antibody Infusion, a report of signal index cases and call for targeted research, published in the American Journal of Emergency Medicine in January 2024. Now, this was a really exciting series of case studies of 3 individuals with long COVID who developed complete remission of their long COVID symptoms following an infusion of a monoclonal antibody called Regeneron, or, my pronunciation is never good of this, casirivimab stroke imdevimab antibody cocktail. All 3 patients had severe fatigue and brain fog in common, but also had a host of other long COVID symptoms. Nancy, could you please tell us first what a monoclonal antibody treatment or infusion is?
Absolutely. So mono means one, just one antibody. Your body makes hundreds of thousands of antibodies every single day. And when you have an infection, it will mount an immune response to all the different parts, bits and pieces of a virus or bacteria or whatever you're infected with. So I like to use HIV as an example because we did so much work on that back in the day, but you might have 32 different antibodies that would be made to that one virus, right?
Oh.
So with COVID, there's the obvious antibody you'd like to have is to spike protein because the virus has that. It binds to the cell with that particular spot, and trying to interfere with that, and better yet, grab that virus and get rid of it is the goal. So one of the antibodies in that monoclonal mix was a spike protein antibody. So that's what monoclonal antibodies are. It's not the same as your own body's immune response, like I say, because it's just one antibody. But on the other hand, it's a whole lot of one antibody, a lot more than your body might have made. It's not exactly what you're doing with your own immune response. It's a much bigger bang for the buck there.
Okay. So how was this treatment used pre-pandemic and then during the pandemic?
It's important to understand that this virus changed a lot and continues to change a lot. These are called variants, right? In the beginning of the pandemic, we had alpha, the beta, and then delta. You might remember that. It's A, B, C, D. We're on Omicron now. And even amongst that, there's been dozens of variants of that particular variant. Your immune response has changed as the virus has changed. And so this Regeneron was created during alpha, beta, delta, and it was approved during that time. And it was being used to treat acute infection. And there was a similar one called Evishield at the time that was being used for chronic infection because it lasted 6 months. They fixed it so that it would persist. That was an AstraZeneca product. So this particular case series, Regeneron, was used in this particular physician's practice. Again, not mine. I got called in, the academic called in later to try to see if this amazing observation could move forward. What happened in these 3 cases was that the patients came in with long COVID with a lot of nerve symptoms, and the doctor gave them Regeneron. First time because they got another case of COVID and they were treating the COVID. And when they got over the acute COVID, their long COVID was gone. So that was the first time. And that encouraged this clinician, Dr. Shepke, to keep using this product in his clinical practice. And then the first 3 clean cases that he got, he told his good buddy who tracked me down and told me about it, and another doctor, and Dr. Marty, and she wrote up the case series. And they bugged me to try to create a clinical trial around it. So we hustled to do that. And we had a problem because alpha, beta, delta, that phase of the illness was pretty much over by early 2023. And so Regeneron would have been effective, one would imagine, for cases that started back then and not afterwards, perhaps. And so we rushed to try to start a Regeneron trial. By the time I got it to the point where I could have submitted it to the FDA, the Regeneron was expiring and we couldn't use an expired product, illegal and bad for you. Don't know what's in there. We went to the company and they were not interested in creating more product or changing the product or working with us, basically. They basically gave us a hard no. So then we went fishing for a new partner. And AstraZeneca, who made that Evishield product I mentioned before, the long-acting product, had created a new long-acting product, later versions of the virus. They were willing to talk to us, and they had product. It was a bit of a process, but I actually wrote the protocol and submitted it to the FDA just about 30 days ago. The FDA has been amazing. They did a rapid review, and I just got the last little bit of information they needed into them today at noon. Had to do it by noon today, but that means we should have our FDA approval in the next 30 days. And so get it to the IRB, and I intend to start this study in January of 2025.
Oh, that's
Again, yeah,
amazing.
very
That,
exciting.
that is
Yeah,
hot off the press.
Doctor, that's really, yeah, it is hot off the press because I wasn't allowed to talk about it until the FDA said it's okay. But I'm assuming that this latest letter means I'm good to go. So
Oh,
I can talk
that's
about it.
just fantastic. I'm so
Yeah,
pleased. I
very
can't,
good news.
yeah.
Dr. Duffy did a lot more cases after those first 3. I think he told me that he did 21 or so and that 19 responded. So that was a very encouraging thing. Again, with Regeneron, not with this AstraZeneca product, but they're very similar in terms of the target. And actually, I'm kind of optimistic with AstraZeneca because this product persists for 6 months. So it gives the chance for the antibody to really clear it out and then keep on clearing it out until it's good and gone. So knock on wood that this spike protein focused antibody is sufficient to clean it all up and help clear it out. We'll see.
Oh,
We will
oh,
find out.
that's
Yeah.
just so exciting. And thank you for sharing that with us. And I can't believe Regeneron didn't want to work with you. It's a possibility that there's 400 million people with long COVID in the world.
I was disappointed. They were encouraging me for a little while, but then they sent me a hard no letter. And honestly, at that point, there were 3 products at the time. All of them were expiring in a matter of a few months or were just expired in spring of this year of 2024. All the products were either the shelf life was over or they would be over by August. And I thought by the time we get this to the FDA, and why get your hopes up and then have a company that's not willing to keep on going? So instead, I went to AstraZeneca with some great help from some really good people to introduce me to the right people. And the AstraZeneca people have been amazing. They've been really good. The other thing is that it's an unusual study because first it's funded by the state of Florida, which is very odd. They're not usually in the clinical trials business. Partnered with the Schmidt Family Initiative for Long COVID, which is an amazing foundation that's doing really cutting edge and innovative work. So they basically are matching each other. It's a matched award to allow me to do this study. So it moved from being a little 20 patient study that would just, show promise to being a proper placebo-controlled trial with 100 people. So
Well, that's fabulous. That is
yeah, it's really
absolutely
exciting.
fabulous. Thank you so much for doing that.
We're very
Uh,
excited. And,
yeah.
it's our top priority. It's very hard to get things like this done quickly. But the
Uh,
FDA, 30 days, they were just great. I mean, honestly, the FDA did their job and did it really well. My team has got a lot of things that we do. We are the clinical trials consortia for Gulf War illness for the country. We home base all of the Gulf War illness related trials that the DoD are funding. So we already had the infrastructure to roll out another kind of study. So we were the right place, the right people to pull this off. Of course, we're very passionate about it. That helps a lot. So I'm able to brag on my amazing team who
mm,
have done some tremendous work. Dr.
mm.
Chima, well, there's lots and lots of great people in our group that have put everything else aside, or at least put it down long enough to to get this thing done. This is a lot of work to do in a very short window.
Yeah,
So
and it's just going to provide such hope to so many long haulers, and it's just absolutely amazing. And thank you so much for sharing that with us. With reference to Appendix A of this paper, Nancy, could you please define what you think are the various categories of long COVID or the post-acute sequelae of SARS-CoV-2 infection and why this is relevant?
this is really Dr. Shepke's baby. I want to give him a lot of credit because he's got a great big job. He's the second in command for all of the public health service of Florida. And yet he, still has a private practice and he did all of this important work. So I'm going to give him the credit that he deserves.
Rightfully so.
Yeah,
Um,
he did some great stuff. There's a group of people that during acute COVID had damage to organ systems that were from the acute infection, permanent structural changes from the acute infection, and have persistent inflammation and persistent thrombosis types of changes. Then a second group that had the effect on their peripheral nerves, particularly smell and taste. Those people often have just this one complication. But then there's the group that we see as long COVID, which is the focus of this report. It looks more like what I tend to see as ME/CFS, myalgic encephalomyelitis, with the post-exertional malaise and the more systemic symptoms that are seen day by day in our long COVID patients.
With reference to Appendix B, can you explain why you think the monoclonal antibody infusion had this miraculous effect? What do you think is a pathophysiology, i.e., the scientific function changes occurring in the body that could result in this remarkable outcome?
So really, without going back to the appendices, but more what I've learned in the years since, because there's been some really important work about COVID persistence in the past year. So remember, we wrote this paper like a year and a half ago. One is this very important work by Dr. Ware at Harvard, who's got one of the best assays for viral persistence, or at least antigen persistence. In fact, he's going to be doing the assays for our long COVID study, for the monoclonal study, which I'm grateful to him and to the people in his laboratory, is they developed this assay that's able to pick up very small signals and help us understand. They published a paper about a month ago that said 42% of people with long COVID still have persistent spike protein antigen that they can measure. And that's the first time it was that high. There was a paper earlier about 25%, and then some very small papers that were picking up between, say, 20 and 40%, but with really tiny numbers of people in them. So that was a really important paper. There's a paper by a Chinese group that if people came to surgery for some completely different reason, they did biopsies of various tissues. And they found a very high level of persistent spike protein as the measure of something going on related to COVID in the tissues, in the organs, far higher than what they were seeing in the blood. So it's possible that the bloodstream's not even the best place to be looking for whether or not there's virus persisting, and it seems to be more in the organ systems. And then that paper that we talked about by Dr. Vust in Amsterdam, he did muscle biopsies and he looked for nuclear capsid antigen, which is different. That's not the spike protein. It's another part of the outside of the virus, but it's a different part. And he found it universally in his controls as well as in his sick patients. And his conclusion was that he couldn't blame COVID persistence for what he was seeing in those muscle biopsies because he saw it in the controls as well as the sick people, but that everybody had it. And that is disturbing because there's another presentation just a few weeks ago by Dr. Fraser in Canada, who's also funded by the Schmidt Family Foundation. And they showed in a metabolomic study that they could tell sick people, long COVID, from recovered people, acute COVID, but they could tell people who never had COVID from recovered people, what would suggest that you've ever had COVID, you still have a signature, an epigenetic phenomena, something happened that stayed with you even after you recovered, that you can pick up in a metabolomic study. So, wow, that's spooky. Okay. I mean, that's saying, since most of us have had COVID at this point, what's the percent? I don't know, but it was high 80s last time I looked. Most people have seen COVID, and we still have lots and lots of COVID in our communities. We have a great outbreak going on right now. That there's permanent changes having ever had COVID. And so then when you say, well, what about this monoclonal antibody of yours, Nancy? My answer is, we're going to see, because spike protein to me is the most damaging part of the virus. So it's possible that people with long COVID have persistent virus. That would mean they're still replicating virus and making virus and it's still attacking stuff. A little hard to say that's for sure what's really going on if we're not seeing it in the bloodstream that much, but we're seeing it elsewhere. It's a little bit, I mean, not impossible to postulate that, but you're thinking it's in some compartment somewhere, not necessarily in the bloodstream, if you're seeing it universally in tissues, but not universally in blood. There's something called compartmentalization. Where does it go? It's like in HIV. When we completely clear HIV, because the blood-brain barrier is so good at keeping the antivirals out, you could still have a reservoir of HIV in the brain that never got to see the antiviral. That would be a compartmentalization that's in the brain, nowhere else. That's HIV, not COVID. So we're sitting here looking at COVID going, where's this damn virus hanging out and how do I get there? That's one thing. Then the question is, will a monoclonal antibody against spike protein clean it up? Well, it's very exciting that Dr. Shepke's patients got so well. That definitely makes you think maybe so. There's another possibility, which is that the message RNA for spike protein is been given to some other cells, not necessarily the whole virus, but just the message to make this spike protein. So there's two possibilities, that there's a whole virus replicating, hiding out somewhere, not necessarily the bloodstream, but hiding out. And there's the other possibility that it's not a whole virus. It's the message that makes the spike protein, and you're swimming in spike protein antigen, which really would mess you up, because the ACE receptor, which is everywhere in every organ system, but particularly on the endothelium of your blood vessels, it would trigger an inflammatory response that would promote the thrombosis and the microclotting that we see so commonly in long COVID. So it could be that spike protein's like the first start, trying to go upstream from all the downstream stuff that makes you sick. That might be the very spot that we should intervene in, is try to get that spike protein out of the system. Or it could be that we have actual whole virus and we're trying to clear it out.
Yeah, I think your hypothesis coincides with a lot of Amy Proel of Polybio's work. We interviewed her and she's all about viral persistence and how the virus
So
is
important,
hiding
yeah.
in organs.
So
So
I'm hoping monoclonal will do it. I mean, say Dr. Shepke had this amazing response to monoclonal. So you got to say, if you got a case series of 19, let's go try that out. But sometimes, as we learned in HIV, just getting the viral burden down for a while gives the immune system a chance to recover and do its darn job, right?
wow.
So it could be that it's a 1-2 thing instead of that you're just the one thing. But we'll find out, right? And then we're going to measure everything. I'm making a biorepository. Poor patients that volunteer for this study are giving me a little bit of almost everything they got so that we can better understand what's really going on.
Well, Nancy, that is just so fantastic. Thank you for an exciting and stimulating set of interviews. And thank you for sharing this hot off the press news with us about the amazing study you're going to do into monoclonal antibodies. I'm so glad that you've had that FDA support. Thanks to you and all those in your team for providing hope to individuals around the world suffering with complex chronic disease.
Thank you. And if you would put it in your links too, we have our own podcast series. We have some amazing information. It's also got the word hope in it, hope and help. It's very much focused on giving people the pearls of information that's clinically relevant to you, the patient, that makes you feel more hopeful and perhaps give you some tools to help with your own struggle to get back to health.
Yes, I will do that. I've been listening to some of them, and yeah, we'll put it in the show notes, and we'll probably feature it on our website too as one of the useful podcast information series. Thank you again, and I'm saying to the audience, please join us next week for another episode of Long COVID: The Answers. Some questions for viewers and listeners to consider. What are your top 5 takeaways from this episode? How will this episode change your practice or perception of this disease? What will you do to act on what you've learned? Please discuss your thoughts on our social media outlets, Twitter or X, the website, blog, Instagram, Facebook, or LinkedIn. Please rate this episode.